Evaluation of media. For this exercise, you will be comparing the growth of two different bacteria in 4 different types of media. 2 of the media are what are known as defined. The other 2 media are known as undefined, or complex media. To begin, label each tube with the name of the media that it contains and which bacteria will be placed in it. Inoculation of each of the media can be done using a serological pipette or a micropipette. Shown first is inoculation with a serological transfer pipette. Each of the tubes will be inoculated with 1/10 of a milliliter of culture. The most efficient way to use the serological pipette for transfer of the bacteria to each of the cultures is to draw approximately 1/2 of a milliliter of culture into the serological pipette. You will then transfer 1/10 of a milliliter into each of the tubes. Shown here, the culture is being drawn up into the serological pipette. Notice, from the 0.3 to the 0.4 is 1/10 of a milliliter. After you have drawn up the culture into the serological pipette, you can put the stock culture away. You will now use the pipette to measure 1/10 of a milliliter of solution into each of the different media. The same pipette is being used for all 4 of the media. This is a very quick and efficient method of inoculating the 4 different broths. Another way to inoculate each of the broths with 1/10 of a milliliter is using a micropipette. Be sure to set the micropipette to 100 microliters, since 100 microliters is 1/10 of a milliliter. When using a micropipette, you should practice with the pipette first before actually using it for inoculation. You may wish to ask your instructor how to use the micropipettes that are in your laboratory. Once the micropipette has been set to 100 microliters, put a clean tip on the end of the micropipette and obtain your inoculum. You will then aseptically transfer 100 microliters of your stock culture into the first tube containing one of the media. After completing the inoculation, dispose of the tip in biohazard bin. A new tip will be used for the next inoculation of the next broth. The tip of the micropipetter for this particular type of inoculation should always be changed between each bacterial transfer. Use of a micropipette ensures that the exact same volume will be inoculated into each of the 4 broths. Repeat this procedure for the following 3 tubes. Using a spectrophotometer to determine growth. A spectrophotometer is used to measure light absorbance through a medium. You will be comparing absorbance values between 4 different media: yeast extract broth and nutrient broth, which are complex or undefined media, and glucose salt broth and inorganic salt broth, which are defined media. Organisms capable of growth in complex media are often known as fastidious. These are organisms that rely heavily on their environment for their carbon sources. Fastidious organisms are incapable of growth in chemically defined media such as ISB or GSB. To begin using the spectrophotometer, you must first set a blank for each of the media you will be testing. Here, the blank for GSB is being used. Be sure to wipe any fingerprints or dust off the tube using a chem wipe. Insert the tube or blank into the spectrophotometer and press the 0 ABS button. Wait until the spectrophotometer reads 0.0 A for absorbance. Once the blank has been set, you are ready to begin recording absorbance readings for each of the bacteria growing in the GSB broth. Again, be sure to wipe any fingerprints or dust off the tube. Place the tube into the spectrophotometer and wait for the reading. Shown here is the absorbance rating for E. coli in GSB. Now you will take a reading for the Strep sanguis growing in the same media, GSB. Again, wipe the tube, place it into the spectrophotometer, and wait for the reading. Sometimes you have to wait for the reading to stabilize. Here, Strep sanguis gives an absorbance rating of 0.328. This process will be repeated for each of the media and each of the bacteria growing in those media. Using a spectrophotometer and recording absorbance values for each of the bacteria growing in different media will allow you to differentiate between fastidious and non-fastidious organisms based on the density of the cultures as seen in the absorbance readings. This is a relatively simple test to use to determine the best growth requirements for bacteria for culturing and identification.